We okay. Um, we were initially funded by a 2-year ARA grant, a so-called Obama grant, back in 2009 for 2 years, which allowed us to set up a registry and biorepository, get the infrastructure set up with 9 sites scattered across the United States plus 1 in Canada. We were fortunate to get a 3-year U54 and get back in sync, and have had 2 more cycles of 5 years. We, in that period, expanded, doubled the size of our consortium to 18 sites. Actually, we had one that we dropped out and added one, and along the way as well. So it was not just simply doubling. This is a map of where our sites are. We have currently 1 affiliated site because the PI there retired, and but we do want to maintain the patients that that person recruited for the registry. Sorry, I'm getting some background here. Okay, thank you. Okay, so here's more background about what we're currently doing. These are our current major research projects. Our first one is the clinical registry, which we started, as I said, with a 2-year ARA grant. And the link to that is a repository, and it's also a longitudinal study. We're supposed to do yearly follow-ups on all of the patients. Next is, since we're still defining mitochondrial disease, we're doing next generation sequencing of genetically undefined subjects. And then the third is the do functional analyses. So we're still in a discovery phase of mitochondrial disease, and it's still an important area. So that's why we're focusing on the registry and genetically identifying causes, and then proving that some variants of uncertain significance are pathogenic by doing functional studies. Our fourth project is on a genotype-phenotype analysis of pyruvate dehydrogenase complex, and linked to that is a newborn screening program. And that's also a longitudinal study. And finally, we have what started out to be an expanded access treatment study of TK2, thymidine kinase 2 deficiency, one of these rare autosomal recessive mitochondrial disorders. But we were told by the FDA at one point, just before we started this last cycle, that we shouldn't enroll additional patients into that expanded access, because they wanted patients to be in a industry standard data collection study. So we quickly pivoted to an activity meter study. We have pilot projects and a career enhancement program, and I'll describe a little bit about those. So really, the foundation of our consortium is this registry. We have 2,100 plus subjects enrolled. We've exceeded our target of 2,000. As I said, we're still, in the earlier descriptive phase of some of our research, and we've learned that, they're quite heterogeneous diseases, of course, but we learned that it's a bimodal distribution. About a quarter of the patients are infants or less than 2 years old, and about a quarter are adults. And the early onset young patients generally have autosomal recessive conditions, and the adults and older patients have mitochondrial DNA mutations. So we're characterizing them in a broad way. And as I said, a lot of our patients are genetically undefined. In fact, we have these classic syndromes like Lee syndrome, MELAS, Murph, and so on, that we use to categorize patients clinically. But the 2 leading diagnoses in our registry are multisystemic disease and other clinical diagnoses. So they often don't fit one of these classic syndromes, and they're often genetically undefined. Even when we do find pathogenic variants, they're quite heterogeneous. And here, this box plot, we see the most common group of these disorders among the nuclear gene defects is Paul gamma mutations, and then second is pyruvate dehydrogenase A1 subunit gene mutations. What you see here in the tan box is that a lot of our genes that represented in our registry are represented by a single patient. So these can be ultra rare. So they're, and quite diverse. One of the things that we observed from speaking to patients is that they often described how they had a great difficulty getting to their diagnosis. So, and one of the tools we've been able to use is surveys, and that's been very effective. And here are two survey reports we've put out, and they showed that these patients go on these long diagnostic odysseys. And in our first paper, we reported that the patients were seeing on average more than 8 physicians. And the second one was an elaboration on that. It showed that these patients are often seen by primary care physicians, but in the end are diagnosed by people who are subspecialists in neurology, clinical genetics, or metabolic disease in most cases. So they really have to funnel through. And this illustrates one of the difficulties in our field is just getting to the diagnosis. And this resonated with the patients. And, although this seems like an obvious thing to do, and it really had an impact, not just in resonating with the patients, but with our patient advocacy group, who took it on to then set up a free genetic testing program in conjunction with industry support. So I think this had not only an impact academically, but also, practically in terms of a very important outcome that helps patients get to their diagnoses. All right, and then we try to redefine the classic mitochondrial disease syndromes clinically, because as clinicians, we see these patients and we need to recognize them and identify them and categorize them, so to help direct sometimes the genetic testing, but also certainly for clinical trials, we need to understand what are the clinical features of these disorders and how to then set up outcome measures and trials and natural history studies. And this was another important survey for our group. I mean, these surveys, they don't come out in the most high profile or high impact journals, but they do have an important impact. And this is one that was done, led by Zuela Zokipley-Cunningham from CHOP. And she took a survey to see what were the most important symptoms for which patients might participate in clinical trials. What would motivate them to go into a clinical trial? And interestingly, the top 3 are related to myopathy, muscle weakness, chronic fatigue, exercise intolerance. And this had an impact, in my opinion, on the industry participation in clinical trials, because industry took this information and multiple trials have been done in the last 5 to 7 years in the primary mitochondrial myopathy space. So I think that this helped to direct some of the trials, or encourage some of the industry partners to work in this space. Some of the other contributions in our pilot projects, one of the very first pilot projects that we funded was to Marnie Falk at CHOP, where she developed this mitochondrial disease sequence data resource, MCTR. This is a web-based resource that provides information about pathogenic mutations in both nuclear and mitochondrial DNA, and provides a very valuable resource for people in the clinic to look up, these variants that are being identified by genetic reports and looking to see what's known about them. And then the second area, as I mentioned before, after sequencing, we often have to do functional studies, and Johan van Hove has been leading an effort, first funded by a pilot project and then through the NAMDC, one of the clinical research projects, on characterizing cells from patients to identify pathogenic variants. And in this ongoing study of deep genotype and phenotyping is led by Jerry Bedoyan at the University of Pittsburgh. So these are all important projects for us, and have been productive with publications. We've had other publications, sometimes in high-profile journals. We have at least 135 publications linked to NAMDC. Of course, our goal ultimately is to find treatments and cures for patients with mitochondrial disease, and this is an area we've started to work on. There are 0 FDA-approved drugs for mitochondrial disease, so this is uncharted territory, by and large for us. So one of the areas, one of the diseases we've been focusing on is this thymidine kinase 2 deficiency, an autosomal recessive condition that's predominantly a myopathy. And defects in this nuclear encoded gene lead to depletion and deletion of mitochondrial DNA because you need this enzyme to make the pyrimidine triphosphate building blocks for mitochondrial DNA synthesis. And as I said, it's predominantly a recessive. It is a recessive condition, but variable in onset and progression. Those with early onset and often in infancy have the survival curve shown here in the blue, very rapid progression and 50% mortality after a year, whereas those with later onset have slower courses and more prolonged courses and longer survival. So we were able to initiate a therapy that we developed in the laboratory, initially deoxynucleoside monophosphates and then nucleoside therapy for this, and it worked in our mouse model. We were able to take it to patients under expanded or compassionate use protocols, both in the United States and elsewhere, particularly in Spain. And we saw very dramatic effects in many of the early onset cases. And here you can see that in this preliminary report that compared to historical controls in the blue, all 5 early onset cases were alive. And so this was quite significant. And we saw stabilization and improvement in motor functions in many of these patients, as illustrated here by the 6-minute walk test. You can see some of these patients became able to walk much more effectively. But at one point, actually about this time of this publication, and about the time of the onset of this last cycle, we were told by the FDA that we should stop enrolling patients under this expanded access program and switch either to an academic sponsor or industry-sponsored industry standard data collection if we wanted to get drug registration. So we then partnered with a company to transfer these patients from compassionate use to phase 2 trial, which is ongoing, and hopefully will be the pivotal trial because the company plans to submit the NDA later this year. So we then transferred this project to a TK2 activity meter study where we're using this ActiPal device, a very simple device that's taped onto a limb of a patient and allows a recording for up to 2 weeks. We generally record for a week, and we're in the process of collecting this data to go along with the industry-sponsored study. So this is sort of an adjunct study that we're using to confirm that the motor improvements that are being measured by other standard scales. All right, so some other contributions of our group to the field. We have a biorepository. We send blood to the Mayo Clinic for DNA, plasma, and mononuclear cell extraction. And a number of, we have over 360 patients with samples, and a number of academic investigators have used samples from this biorepository. We also have a virtual fibroblast biorepository where we have sites perform skin biopsy locally, store the cells. And this has also been a popular resource that's been shared not only with academic investigators, but also with some industry people who are interested in testing some potential therapies in these cells with these rare genetic variants. And of course, as I mentioned earlier, the registry, it's quite an extensive registry with 127 data items, and that's a lot of information. And we're looking to see if we can prune that down and try to focus this in the future. But in the meantime, we continue to collect data. We've published our first paper on this back in 2020, and that was an important paper to describe what we have in our registry. It was the second most cited paper in that journal that year. And we've developed a data mining tool to allow all of the investigators to look at the data in real time and to see what's, poke their heads in and see what's in there and hopefully use that information to develop research projects. We've planned to try to share this with a global mitochondrial disease registry called Genomit, which is based in primarily in Europe, although other countries across the world, including Asia and and other European countries are planning to participate. So that this global registry, if everyone participates, will have more than 19,000 patients. So that's quite a powerful uh, registry if it can be pulled off. We are trying to share now with a new foundation called the Polymerase Gamma Foundation and try to add our 115 patients from our registry to a growing list of patients that will be published in a retrospective analysis. And we do share this information in the registry with any academic investigator or industry as well, if they're interested in knowing, where the patients are, how many there are with particular diseases. So we think we're trying to serve many constituents in the community here. Briefly about our pilot projects. We've funded 1 to 2 a year. So we've funded 15 different pilot projects, 7 of which have received additional funding, so almost half. And this is the list of the pilot projects. As I mentioned, Marty Hawk's MCPR project was one of the first. One I wanted to highlight here is Fernando Scalia's project, citrulline supplementation as a treatment for nitric oxide deficiency in MELAS. And this is a phase 1 dose escalation study. Sorry, I just wanted to mention that his study is actually just finished enrollment. So we're hopefully we'll complete this first phase 1 study, from design through completion and analysis in this funding cycle. So that he started in a previous funding cycle, but regardless, he'll hopefully finish it. And so this is quite an accomplishment for this rare disease. We learned along the way that pilot projects, particularly for clinical studies involving patients, take a long time to often set up. And so Johan van Hove, who has been leading this program in our consortium, has had to redesign how we administer this. And this is a bit of a complicated slide, but he has formalized this, so we have 3 reviewers for each proposal in July, and we try to turn it around quickly and get the results to the investigators in August or so. And what often takes a lot of time is just getting a study protocol, an IRB protocol written and approved. So we try to get it to them early, the results early, so that they can start to work on that. And a lot of our investigators are young with these pilot projects because they've never written a protocol before. So they need help, and they get help from us and from the DMCC when they review the protocols, and the NIH officers as well. And we try to move things along with quarterly reports, just brief reports, just to check in on how they're doing, and request that they give a final report at the end of the year. So this has kind of helped a bit, but it's still a bit of a problem getting these short pilot projects going. And frankly, they're usually not completed in that year. How has participation in the RDCRN kept junior investigators in this field and even advance them through the course of the training? Well, I think our career enhancement program has been very successful. It's been led by Richard Haas at UCSD, and you can see here the smiling faces of some of our former fellows. 2 in particular, as well as Kipling Cunningham and Austin Larson, were among the first investigators, and they're currently on faculty at institutions. So Zoella at CHOP and Austin Larson's in Colorado. So they're rising stars in the mitochondrial disease community. Actually, all of these people are still involved in mitochondrial disease research. So we're very pleased that this has been a very successful program for us. Sorry. But it does present some challenges. By the time people think about subspecializing in mitochondrial diseases, they're further along in their careers, often as in this case. There may be children involved. It's hard. We originally set this up as a rotating fellowship, but it's hard with people with families, so we had to adapt it and make it available for people to do, not necessarily at UCSD, but perhaps at their home institutions and spend their whole year at an institution rather than the original design. But anyway, we've had to adapt along the way. We also have been trying this career enhancement program, Gateway Grant. It's a small award, $5,000, to encourage junior trainees, whether they're students at undergraduate or graduate levels, to explore mitochondrial disease over a summer or part-time over a year. And we're still in the process of seeing how that's going to work out. One of the questions we were asked to address is what was Consortium able to do since it became part of the RDCRN that it would not have been able to do otherwise? Well, we've established the first clinician-driven disease registry for mitochondrial disease and biorepository. It's the only one in North America. Other countries have them. Because we now have a registry and biorepository, we're able to participate internationally in collaborations that we wouldn't have had we not had this funding in this registry. We've really generated a nice community here of mitochondrial disease researchers across the United States and one in Canada. So I think this is really a great benefit, is to develop this community of people that were previously siloed in their individual centers. We've trained some of the young people to become mitochondrial disease clinician investigators, as shown. I think we've improved the diagnosis of mitochondrial disease in North America, and we've started to foster early clinical trials through the Expanded Access Program on TK2 and the phase 1 study of citrulline and Milos. And there have been multiple pilot and research studies funded through all of this. I think we've also disseminated information about mitochondrial disease. This is data from the DMCC showing the number of hits in the first quarter of this year to the NAMDC website, and we have about almost 1,000 hits altogether in this quarter, and you can see it goes up and down, but some of the things that people outside are looking at are our overall network, the clinical sites involved, disease study, research studies going on. So I think we're getting word out that mitochondrial diseases are being investigated here, and we're moving forward with this. How has this helped our relationships with PAGs? And in particular, we've worked very closely with the United Mitochondrial Disease Foundation, which is the largest one in the United States, if not the world. We have very close relationship with them. We have weekly Zoom meetings with them. They participate in a monthly NAMDC meeting, and we've referred lots of patients directly to UMDF, and vice versa. So we've shared information between the two groups about patients. I think the UMDF now has a better understanding of what goes on in academia and what we're hopefully capable of. UMDF has really supported us financially as well by providing the space and funding for an annual face-to-face meeting in conjunction with their annual meeting, the Career Enhancement Program, and as well they've also partially funded our infrastructure, our administrative structure as well. We've hopefully given them some useful advice too, and helping them to set up a patient-driven registry to complement our clinician-driven registry. So what are some of the lessons we've learned? Well, of course, clinical research is easy to propose, but challenging to implement. We've learned that human subject studies almost inevitably take longer than we anticipate, especially for junior investigators who don't have the experience and have never written a study protocol. And recruitment and implementation for these natural history studies. There have been 4 that have been attempted here at NAMDC, has been much more difficult than we anticipated. These are very rare diseases. It's difficult to capture them and then to hold on to them in natural history studies. We've learned that the single central IRB are difficult to implement, but certainly can improve consortium efficiency, especially for stable long-term studies where the renewals seem to go much more effectively than regular IRBs at multiple sites. Central administration with a dedicated project manager is essential, and hopefully, preferably with a project management team. So we work together centrally, but coordinate things with all of the various sites. And we have to have engagement of the investigators and the coordinators at the sites. It's an awful lot of work to do, and it's important to distribute the responsibilities of leadership. So in particular, our career development pilot projects have been led elsewhere, and that's been very successful. It's important to establish the governance and cooperative agreements early on. And here we were asked to list some of our takeaway messages, and here are a few. Clinicians often underestimate the work required to conduct clinical studies. I think we think that for clinical trial design, simple but rigorous is superior to complex and sloppy. And I think that can be illustrated by our registry, which we collect a lot of information, but it may, the complexity means that not all of the data are being collected and entered in the right way. So we're looking at ways to streamline that. It's important, of course, choose your collaborators who have the bandwidth and the willingness to work. Everyone's going to say they would like to be part of it, but you really have to be selective in choosing the people who really can do the work. And then clinical research experience is clearly important, and we've gained a lot through this funding of our consortium. And we're a lot better off now than before. We had virtually no clinical trial experience prior to the beginning of our consortium, and now we're very much engaged with that. And investigators with experience need to train those and guide those with less experience. And then here's some words of wisdom. I think these are pretty obvious, but you need to set priorities to fulfill the most important and achievable needs. Many things are very important, but it's important to choose your battles and choose ones that you can actually achieve. And I try to tell my colleagues that be realistic and maybe under-promise a bit, but over-deliver on milestones and to make people feel like they are accomplishing a lot, which we are. But it's nice to have that if we can meet our milestones. And then it's important to have regular, clear communications with investigators and coordinators. And it's important to know what your collaborators want and what they're capable of producing and doing. We all want so much, and we just need to recognize that we all have limited time and resources, so we have to keep that in mind. And it's important to engage everyone as much as possible, and obviously heavy lifting is easier with multiple hands, so that's important. So last couple of minutes, I just want to mention what preparations have been made by our consortium after we graduate. So we have begun sustainability discussions with our TAG early last year. We had a sustainability survey among the investigators and coordinators last spring, and we discussed the results at our June face-to-face meeting. We also conducted, our UMDF actually on our behalf, conducted a survey of potential industry partners back in October. And we just had a in-person sustainability meeting just this past weekend on Friday and Saturday, where we discussed the strategies for sustaining our consortium. And then we're hoping to put together a plan in time for our next face-to-face meeting in June. Hopefully we'll begin to implement that plan during the no-cost extension year starting September 1st. And where do we want to go from here? We want to sustain the critical core activities, the registry, biorepository, but we want to move more into natural history studies and clinical trial readiness, which were identified in the surveys as critical in our field right now. And we need to do this not only with support from the UMDF, but we'll have to find research grant funding to support research projects. We need to better define our role in the whole mitochondrial disease landscape. There are a number of organizations and PAGs that are involved in mitochondrial research. We need to find our niche in all of this and where we could be most effective. And that was part of our strategic planning meeting this past weekend. And we want to continue to grow with our UMDF collaborators. And increasingly, we'll want to engage with industry, which fortunately has taken an interest in developing therapies in our area. This is just a timeline. Sorry, I tried to format this, but every time I formatted it, the letters came out screwy. So anyway, I already went through all of this, and I just want to end with a thank you to everybody, particularly NIH for funding us, all the different agencies, the divisions of NIH that supported us, the RDCR, DMCC, all of the people in our collaboration, and of course our PAG. And I thank you for your attention.
Well, Michio, you were the first, but you set the bar high for everybody that follows. That was phenomenal. Thank you very much. I open it up for questions and conversation. Looking at the chat, it looks like it's just singing your accolades right now. I do have a question. A couple of questions myself. The biorepository, does that cost money to maintain? And I assume that's part of sustainability. Maintaining such a precious resource would be first and foremost on your list.
Yes, absolutely. And both, of course, the registry and biorepository are linked, and they both cost money. And so what we do is try to have it. Well, we have the centralized biorepository at the Mayo Clinic, where obviously they have like probably the best biorepository system in the country in terms of just efficiency, and so it was very cost effective to do it that way for us, at least for the blood samples. And we pay sites for each sample that they submit, so we do it that way. It's equitable. Maybe we're underfunding that, and we need to maybe find ways to better support that. But I think that's the way we've managed it. And then the fibroblasts, it was just easier for sites who are often collecting the fibroblasts for their own research purposes to have the virtual fibroblast set up that way.
I had a quick scientific question. In one of your beginning slides, you showed that one of your largest cohorts are patients with multi-system symptoms, but no genetic diagnosis, if I understand correctly.
Yeah.
That, just so I understand, those patients have not had genetic testing, or they've had initial genetic testing and they're negative? If so, what are you planning to do with them?
Yeah, so it's both. Some of them, most patients nowadays have genetic testing. Some of the ones that we enrolled early didn't, of course. So we do have, as Project 2, whole exome and whole genome sequencing. So that's a lot of them are supposed to feed into that pipeline. And then if we get variants of uncertain significance or probably pathogenic, then we try to do functional studies in Project 3. So that's the way we were set up for this discovery. And frankly, the sequencing isn't going as fast as we'd like, but hopefully we can continue.
And before I let Sandesh ask the last question, does whole genome sequencing include sequencing of the mitochondria?
Yeah, and often sometimes we do the mitochondrial DNA separately as part of the next gen sequencing.
Interesting. Sandesh, you have the last question.
Michio, thank you. I wanted you to just advise the rest of the consortium on the policies from NAMDC as to how are you handling the requests for data that come from external collaborators or scientific academic partners, and how NAMDAC is handling some of the clinical trial readiness from industry partners regarding the data for designing trials? What's the process for data sharing look like at NAMDAC? I'm sure we can learn a lot from the policies there.
Yeah, so actually, we do actually need to formalize that a little bit more, but we do have a data use committee that, and we have a form that I'm happy to share that form. It's very simple, asking exactly what their purpose of what they're planning to do. We only give de-identified data. We never give, obviously any dates of birth or names or anything like that, but we do share de-identified data, and preferably we like to give it, an aggregate. So if somebody asks specific questions, how many patients you have with Paul gamma mutations and what are their phenotypes, we will give that information. We don't like to give individualized data because sometimes it could be identified just by the mutation, and certain clinical features might give away who they are. But our preference is to give aggregate data. And then we give this all free of charge, of course, to academic investigators. We don't charge industry except to, if they have to do some work on it, we just charge it for the labor of whoever's doing the data management part, but that's about it.
Thank you.
Well, Michio, thank you very much. We really appreciate you sharing your expertise. That's a phenomenal amount of work, and I got to think the momentum you've got is going to be sustained. This is the answer can't be anything except that.
We hope so too. Yeah, we had great enthusiasm at our meeting, and now we have to move forward with that.
That's excellent. Aaron, do you want to put the itinerary of the agenda back up? All right, perfect. Do we have